The main aim of this research is to monitor the distribution of endogenous histone H3 lysine modification in living cells without disturbing cell growth and development. Histone proteins have to undergo several modifications in order to become functional. These modifications are collectively known as post-translational modifications. Few processes include acetylation and methylation. These steps are important in cell signaling metabolism and also helps to maintain gene expression and genome integrity. In this research, Fabs, Fablem, and monoclonal antibodies were used. Fabs bind to a specific target without causing any disturbance in cell growth and embryonic development. Monoclonal antibodies are specific to only one antigen. Fabs produce distinct nuclear patterns that are characteristic of their target modification. H3K27 trimethylation specific Fabs were used on mouse embryos for this study. In order to study the nuclear patterns, Fablems were used. It is a tool used to track histone modifications which is compatible with simultaneous monitoring of more than one modification. The first step was to generate monoclonal antibodies, MABs. Mice were immunized with synthetic peptide. After generating hybridomas, the clones were screened and isotyped using a kit and purified. The resulting clones were then measured to determine affinity of the MAB. Fabs were prepared according to the manufacturer's instruction. The samples were concentrated up to 1 mg per milliliter and then was conjugated with a fluorescent dye. The sample was then purified and the Fab concentration and dye to protein ratio was calculated. Next, the cells were imaged under an inverted and confocal microscope. Cells were placed on a glass dish with a cover slip and fluorescent Fabs were pipetted on the cover slip. It was then placed under an inverted microscope. Mice oocytes at anaphase 2 and telophase 2 were injected with Fabs and 5 microgram per milliliter of histone and placed on an imaging device, and 51 images were captured at 15-minute intervals.
In Figure 1a, the imaging of living cells of histone modifications with the use of immunoglobulin and Fab is shown. First acetylated histone H3K9 specific monoclonal antibody was used to compare immunoglobulin and Fab's properties. After it was loaded into the cytoplasm of HeLa cells, immunoglobulin was too large for the nuclear pore, so it was excluded, whereas H3K9 as AC specific Fab is smaller, so it diffuses into the nucleus immediately. Fabs appear to be more convenient than the immunoglobulin molecule for living cell imaging, as it has easy access. Validate whether Fab H3K9AC is targeted to H3KAC on chromatin in the living cells. HeLa cells were reloaded and cultured for 4 hours. Fluorescence recovery after photobleaching was done, which is shown on Figure 1e. After loading HeLa cells, it went through several cell divisions, and when the images were compared, it was evident not all H3K9AC was occupied by Fab H3K9AC. Table 2 represents the embryos generated by in vitro fertilization that were injected with Fab H3K9AC 488 together with 5 mg per milliliter of H2B MRFP mRNA. Embryos were then transferred to a uterus after 2.5 days post-coitum in pseudo-pregnant females. With the antibodies injected, it improves the embryo development, and the number of embryos transferred supports this. In Figure 6a, oocytes were fertilized in vitro injected with different fluorescently labeled Fab histones. This showed H3 acetylation specific Fabs were concentrated in pronuclei, whereas methylation specific Fabs were distributed throughout the egg cytoplasm. In Figure 6b, Fablem performed on embryos with somatic nuclear transfer is shown. Oocytes were injected with a mixture of fluorescently labeled Fab and histone H2B, MRFP-B1, mRNA, enucleated and injected with a somatic nucleus derived from a cumular cell. Then reconstructed embryos were activated with SRCL2 and cytokeratin for 6 hours. The embryos were further incubated for 3 hours in TSA, monitoring the histone H3 acetylation levels. Time-lapse confocal images were recorded for IVF and SCNT.
In this experiment, Fablem didn't affect regular cell growth or embryo development, and so this method doesn't disturb normal cell metabolism. This is because the monovalent Fabs only bind their targets transiently. Highly specific antibodies must be used for Fablem since there is no blocking reagent that could suppress non-specific binding, unlike immunostaining for fixed cells. Therefore, monoclonal antibodies are recommended for better control of Fablem experiments. Only acetyl and phosphospecific Fabs were successful for imaging in pre-implantation embryos. This could be explained by different affinities of Fabs. There are several limitations to using this method. 1. The time resolution of FabLem depends on the binding time of Fab. In other words, changes in less than a few seconds can't be traced using a high affinity Fab. 2. It is likely that turnover of modifications is influenced by Fab binding. Therefore, the balance of modifications to demodifications may be disrupted, even though such a change may not affect biological functions of cells and embryos. Nevertheless, this problem can be fixed by using two Fabs with identical specificity, but with different affinities. Lower affinity Fabs should have improved temporal resolution and should lessen blocking effects at the cost of enhanced signal to noise. 3. Fabs continually bind and unbind their target modifications in the nuclei. As unbound Fabs can diffuse into the cytoplasm, the ratio of nuclear and cytoplasmic Fab intensity is linked with that of bound and unbound fraction. Since the nucleus to cytoplasm ratio is subject to both Fab affinity and abundance of target modification, the degree of changes among the different Fabs can't be directly compared. Fabs with a wider variety of epitope binding affinities can be used to observe changes in target modification levels. However, visualization of the modification sites is dependent on Fab affinity and the distribution of concentration of the modifications. When a specific modification is immensely enriched in large foci, Fabs can be concentrated locally. If the modification is scattered at frequent smaller foci throughout the nucleus, then a high affinity Fab combined with a high resolution system is needed. FabLem was applied to monitor changes of H3K9 and H3K27 acetylation in mouse pre-implantation embryos. FabLem showed that both H3K9 and H3K27 acetylation levels are higher in paternal pronuclei. These data suggest that a high level of H3K27AC is important for normal embryo development by antagonizing the methylation on the same lysine residue. Lower acetylation levels observed in transferred somatic cell nuclei might create an abnormal epigenetic landscape, which in turn might cause abnormal gene expression profiles. Increased levels of H3K27AC by TSA may therefore be a factor that adds to the usual development of SCNT embryos. The global level of transcription in two-cell SCNT embryos was shown to increase upon TSA treatment. In comparison, H3K9AC doesn't seem to have much antagonistic effect on the methylation in zygotes.
Besides FabLem, real-time measurements of protein modification levels in living cells can also be achieved using FRET-based sensors. Although a direct comparison of these two methods has yet to be made, FabLem generally has the following advantages. Endogenous modifications can be controlled. 2. Standard single-color filter sets can be used for fluorescent imaging. 3. Many antibodies are available for simultaneous detection of different modifications in the same cell. And 4. No genetic or recombinant material is introduced, therefore imaged cells and embryos can act as donors for reproduction studies.
FabLem also has the following disadvantage. 1. The requirement of protein loading into the living cells by use of microinjection is monotonous, and other methods are not as standard as DNA-mediated expression systems. In the future, many different and easier techniques may be developed which would make this method easier to carry out.
In conclusion, it is expected that FabLem will be a powerful tool for monitoring the dynamic changes of epigenetic modifications during the cell and developmental cycles, and in response to stimuli and small chemicals. The method can be used to visualize any endogenous protein modification, providing a novel strategy to understand the dynamic nature of cell regulation and testing the response of patient cells to potentially therapeutic drugs.